Today I'll be modeling and analyzing the impact of bottleneck effect on a hypothetical cheetah population. I chose to use cheetah as an example because cheetah is a species that has previously experienced two bottlenecks effect and is now currently listed as a vulnerable species by the International Union for Conservation of Nature. Before analyzing the model, we must first understand what exactly is the bottleneck effect. The bottleneck effect is a form of genetic drift which occurs when the size of a population is severely reduced. This can in turn affect the distribution of genetic diversity amongst individuals within the population. Most of the time, the bottleneck effect causes a decrease of genetic diversity, therefore it causes allele to be lost and other alleles to be fixed. The bottleneck effect can also cause evolution because when a significant amount of genetic diversity is lost, the new population will be genetically distinct from the original population, which could lead to the evolution of a new species. Because of this, in order for a population to remain in the Hardy-Weinberg equilibrium, the bottleneck effect must not occur. The question is the cheetah population evolving after experiencing the bottleneck effect? Will help us understand if the change in one of the Hardy-Weinberg equilibrium factor is enough to cause the hypothetical cheetah simulation to evolve. To answer that question, I came up with a null hypothesis and an alternative hypothesis. The null hypothesis states that the cheetah population does not show any significant changes in allele frequency after experiencing the bottleneck effect. And the alternative hypothesis states that the bottleneck effect significantly changed the allele frequency of the cheetah population. I created a spreadsheet model that will help me to investigate further. The spreadsheet will create a first generation with random allele frequencies. Then the second generation will randomly inherit zygotes from the first generation. The third generation will inherit from the second generation, and so on. This will help to simulate the passing of allele combinations through generations. The population starts with 500 individuals, and the bottleneck effect occurs after the third generation. A random event will cause only 20% of the population to remain. In order to observe the impact of bottleneck effect on genetic diversity, I assigned two alleles to the population. Dominant allele A, which codes for largest spots pattern on the cheetah, and recessive allele B, which codes for smaller spots. After running several trials, I noticed a common trend. The bottleneck effect would often cause the loss of recessive allele and the fixation of dominant alleles. For example, after the bottleneck event has occurred in this trial, as the generation passes on, less and less recessive alleles are being inherited. Eventually, the homozygous recessive individual no longer exists in the population. There are 19 homozygous recessive individuals at generation 4, and the recessive allele frequency is 0.335. Compare that to the 10th generation. The table shows 0 homozygous recessive individual remaining, and the calculated recessive allele frequency is equal to 0.01. However, as the recessive alleles are decreasing, the dominant alleles are increasing. There are 45 homozygous dominant individuals at generation 4, and the dominant allele frequency is 0.665. But at generation 10, there are 98 homozygous dominant individuals, and the dominant allele frequency has become 98.01. Although the recessive allele isn't completely lost and the dominant allele isn't totally fixed, but after 1 or 2 generations, the recessive allele will be completely lost and the dominant allele will become fixed. Here are the results of some other trials which had similar outcomes. Although this is the most common outcome, there are several others which I found very interesting. First, there is this trial where the complete opposite occur. According to the graph on the right, which shows the different allele frequencies, the data point for the two alleles are at opposite end of the graph. The dominant allele has the lowest value, while the recessive allele has the highest value, which means in this population, the dominant allele is lost while the recessive allele is fixed. Here is another outcome where the population becomes completely heterozygous. As you can see on the allele frequency graph, the data point for both allele at generation 10 have the exact same value. This is because all of the population has become heterozygous, which means there will be equal amount of the dominant and recessive alleles. Despite having different results, all of the trials have one thing in common, and that is the bottleneck effect has caused the allele frequency of the trial population to become distinct from the original population, therefore indicating the occurrence of evolution, which is not supported by our null hypothesis. In order to find out if we could accept our null hypothesis or not, we'll be comparing the individuals in the 10th and 1st generation by using chi-square analysis. Since the 1st and the 10th generation have different population size, we must first find the percentage of individuals and put them in two equal sized populations of 100 individuals. We will use the 1st generations as expected result and the 10th generation as observed result. Then plug all of the value inside the chi-square equation. After some calculation, we are able to determine that the chi-square value is equal to 161.74. Since we have 3 different allele combinations, our degree of freedom must be 2. And if we're using a significance level of 0.5, then the critical value will equal to 5.991. 161.74 is larger than 5.991. Therefore, we are able to reject our null hypothesis. Which means the cheetah population does have significant changes in allele frequency, therefore indicating that evolution has taken place. Given all the data and analysis, we are able to answer a question. Is the cheetah population evolving after experiencing the bottleneck effect? According to our experiment, the bottleneck effect can cause changes in the allele frequency several different ways. The change it causes is often so significant that the allele frequency becomes distinct from the original population, therefore causing it to evolve, making the population unable to maintain the Hardy-Weinberg equilibrium.